Just to start out with a few acknowledgements. I've learned from many people in this area. A few of them are listed here, in particular Mertosh Babadi, who is the group leader in the group that I'm a part of in the data sciences platform. I've learned a ton of things about single cell RNA sequencing and data analysis in general from Mertosh. As well, I would like to acknowledge Patrick Eleanor and his group in the Precision Cardiology Lab. There are several talented single-cell RNA sequencing focused analysts in that group who I've learned a tremendous amount from. Mark, Carolina, Secundar, and Amelia in particular. And I've worked on several projects together very closely with Mark in the past. He knows a great deal about single-cell RNA-seq analysis. So make sure to ask him a question if you see him around the road. Okay, as an overview of this talk, I will give a brief review just to refresh what I'm talking about, and hopefully we can spur some of the same questions that I didn't leave enough time to answer last time. So I'll start with a brief review. But then I'll focus on some different areas than I focused on last time, in particular things I didn't really have time to cover in very much detail. So the first topic will be about batch effect correction. We'll talk a little bit about that when you have multiple data sets, what to do. Another topic we'll touch on will be subclustering, which is kind of a common task that many people are interested in, but it is a little bit tricky just because it's not often very well defined. Then we'll talk a little bit about differential expression testing, and I'll give sort of my favorite toolkit for doing that, although there are many. And at the end, we can talk a little bit about considerations for experimental design. Okay, so I'll start with a brief review, just so that we're all on the same page, and please do feel free to jump in with any questions. Basically, data processing for single-cell RNA-seq data looks something like this. You start with FASTQ files, or even with BCL files that are right off the sequencer, and you just have a big list of sequences. Somehow you want to go from that to what's on the right there, an interpretable map of all the individual cells in your data set. So it's a big plot with a lot of dots. Each dot is an individual cell, and this two-dimensional representation shows something about how cells are related to each other. So cells that are more related are grouped together more closely in this representation. And when we perform some kind of a clustering, enabling us to give each cell a color, we can see that different clusters of cells grouped together have the same color, and the interpretation of this could be that each color corresponds to a different cell type in our experiment. And once we have a map like this, we can really ask a lot of questions like, what makes that purple cell type number 4 different from all the others? Which genes are upregulated in that group of cells? And we can ask all sorts of targeted questions. So just to get on the same page about where this data comes from, as a brief refresher, I won't spend too much time here, but happy to answer any questions that I can. One particular instantiation of this experiment is the one done by 10x Genomics, and it looks something like this. It's a droplet-based experiment, and the idea is to capture individual cells in their own tiny droplets. So here we have 3 cells, each one in its own droplet, represented by the black circle. Now, the point of having each cell in its own little droplet is that you can do chemistry that's specific to each droplet. And by that, I mean the following. What we can do is break open each cell within its own droplet, capture all of the polyadenylated mRNA transcripts that were inside that cell, copy them to cDNAs, and in the process of copying that mRNA to a cDNA copy, we add 2 barcodes that are made of DNA. And those barcodes can tell us about which droplet each molecule came from. So in particular, there's a droplet barcode and a unique molecular identifier, which is totally random sequence. And the idea is that the droplet barcode is that very same sequence for every molecule that originated in this droplet. So as you can see, all the molecules from this droplet, from this particular cell, got this same orange droplet barcode. So we can tell where all of these molecules came from after we do sequencing. Likewise, we have a unique molecular identifier here, which is unique for every single initial molecule that we captured. We can use that to try to remove PCR biases after we have the data set. So we perform lots and lots of PCR to make tons and tons and tons of copies of those molecules that we barcoded in their droplets. Perform lots of PCR, grab a bunch of molecules and sequence them. And after we have a lot of sequences, we can map those to a reference genome, a reference transcriptome. So for example, here this dotted line represents the genome. The green blocks represent annotated genes. So gene A here, gene B here. We can map all of these reads to this transcriptome and count the number of reads that align to each annotated gene. So here, for example, we can see a group of reads that align to gene B, and each of these reads again is tagged with barcode information about the particular droplet that it came from. And the idea then is to demultiplex all these reads by barcode back into the initial droplet that the reads came from. So we can sort all those pileups of reads by droplet and say, droplet number 1, gene A, these are all the reads that map there. Droplet 1, gene B, these are all the reads that map there. And as we discussed last time, the strategy is to count the number of unique molecular identifiers. So these random barcode sequences, we want to count the number of unique ones that show up, because that's a proxy for how many unique original molecules we found within each cell. Then we can go ahead and enter these integer numbers into the corresponding spot in the count matrix. So the count matrix is droplets by genes, and in each entry of this big matrix we have an integer, and a lot of the time that's going to be 0. So last time we talked a little bit about what the data sparsity means and how to do data analysis when your data set is very sparse.
Stephen, we have a question.
Please.
And the question is it possible for 2 cells to be encapsulated in a single droplet? And if so, or how can we distinguish these droplets from droplets formed by a single cell?
That's a really, really great question. So yes, basically, I've been working on biological data for several years now, not that long of a time, but everything that I've learned indicates to me that if you can imagine a scenario that seems at all plausible, then it probably is happening. It's just a matter of to what extent is it happening. So as the questioner imagined, maybe there could be 2 cells in a droplet, And indeed, there's no reason why there can't be two cells in one droplet. And because it's possible, indeed it does happen. Now, the idea in the experiment is to try to capture individual cells in single droplets. So while we know that these doublets do occur, the experimentalist will try to reduce the number of doublets to the extent possible by really by loading fewer cells into the droplet making machinery, which in this case is a microfluidics device. So what ends up happening is there's a trade-off between we want to sequence a lot of individual cells to get the most data out of our experiment. But if you try to load too many cells, then you can end up getting these doublets. So there's a couple of things to keep in mind as you do this experiment. One is you don't want to load too few cells, in which case you'll see a bunch of empty droplets. And there's really not much fun in sequencing a bunch of empty droplets. So you want to load enough cells that you can see a lot of droplets capturing cells. But at the same time, if you try to push it too high and see, say, maybe 30,000 cells in your experiment, then you're up in the regime where a significant fraction of those could be doublets. Now, there are some computational approaches and some experimental approaches, actually, to try to mitigate this problem. Let's see. So one computational tool that I have found particularly useful for this is called scRublet, scRublet. That tool is useful for trying to computationally infer what might be a doublet in a particular experiment by taking a look at the populations of different cell types that it thinks are there in your experiment and tries to explain droplets as a combination of two different cell types. So in practice, that can be pretty useful. It's difficult to get rid of all the doublets that might be occurring. So that is always something to keep in mind. And there are other experimental approaches to try to mitigate this. I'm not really an expert on those. But the idea is something like using an extra piece of information, some sort of a tag that you can add so that if you see two of those tags in the same droplet, you know that a doublet has occurred. So there are a couple different ways of doing that. And I'm not really an expert, but I think in the future experimental strategies to reduce the doublet rate could also be very helpful. Okay, so just to review the basic workflow, once we had the count matrix, so the basic workflow has sort of three pillars. Per cell quantification, and that's what we've discussed so far, and we went over last time in quite a bit of detail, going from fast Q reads to a full count matrix. Then once we have this count matrix, we begin this second pillar of data analysis, the map making process, which involves cell QC, as we discussed last time, potentially integrating multiple data sets, doing dimensionality reduction, visualization, and clustering to create a map. And then in the third pillar is lots of downstream possibilities for analysis. And so this per cell quantification, in order to get the BCL files from your sequencer into this FastQ format, you can use a tool like Cell Ranger Make FastQ or the equivalent. That will give you FastQ files. From there, you can use a tool like cell range or count or Alevin, or there are many others, which will enable you to go from fast Q reads to this count matrix as we discussed. During the map making process, as we covered last time, dimensionality reduction is typically done through a combination of highly variable gene selection and principal component analysis. And then after you have these principal components, the visualization procedure is a process of taking that reduced dimensionality representation of the data and reducing it even further to 2 dimensions. And this can be done pretty well by either UMAP or t-SNE algorithms in order to get some kind of a map like this. And lastly, clustering to sort of assign colors to all of these dots, which can be done by creating a graph and using an algorithm to partition that graph into different groups. 2 algorithms that are particularly good for that are the Leiden and the Louvain algorithms. So just a review. And as we discussed last time, just to refresh your memory, we added 2 extra steps into this most basic workflow. Those extra steps involve trimming the reads before creating the count matrix, and that read trimming can be accomplished with a tool like CutAdapt, which is the one I like using for that purpose. And then after creating this count matrix, it's very often quite useful to try to clean up that count matrix to remove some technical artifacts. And so last time I talked about this tool that I am one of the authors of called Cellbender Remove Background, that can take a raw count matrix and clean up some of the technical artifacts, including ambient RNA, and leave you with a cleaned up count matrix, which you can use downstream in this map making.
Stephen, previously there was a question about sort of best practices for using Cellbender, and in particular, the attendee last time wondered whether it's typically you would recommend using it in place of general QC filters or in addition to them, and if so, at what ordering in the process?
Yeah, thank you for that question. That's a very important point. So the recommended workflow would be to obtain your raw count matrix using a tool like cell range or count, for example. And then directly after you have this raw count matrix, without touching it at all, actually Cellbender Remove Background, that tool actually requires you to have done no extra processing on that matrix. So you have all the empty droplets included, everything is included, just totally untouched count matrix. You can feed it into this tool and it will produce a cleaned up count matrix that can either be filtered to non-empty droplets by the tool, or it can still be the entire count matrix, including the empty droplet barcodes. So this is just directly after you've done something like cell range or count, you can create this cleaned up count matrix. But then at that point, as the question alludes to, it's very important to still do this cell quality control procedure. So you can proceed with this kind of cell QC in just the same way you would have done with the raw count matrix, but you still apply cell QC procedures to this clean count matrix in the same way. So the idea of this kind of quality control step is to get rid of maybe dead or dying cells, cells where you really didn't capture a lot of reads, doublets, things like that, for example, that can really confuse the final map that you make. So yeah, the intended workflow is to run this tool directly on the raw count matrix and then still do all of the same quality control procedures you would have done.
Great, thank you. There's another question about Cellbender that's just come up now, and this, the attendee mentions that it's a technical question, and the question is, if the Cellbender removes a background plot does not converge and it's very spiky after adjusting the learning rate a few or several times, is it okay to proceed with the output matrix, or should some other approach be used?
Yeah, that's a good question. So, as part of developing this Cellbender remove background tool, I've had the opportunity as part of the precision cardiology lab to run this tool on hundreds of data sets so far. And we've done some work with Aviv Regev's group as well on a couple hundred more data sets probably. And so we've seen quite a few different things that can happen. And there are cases where there are some difficulties with convergence of the tool. So what it's doing is, under the hood, is it's learning a probabilistic generative model for the data itself. And as part of that model, there's counts that come from cells and counts that come from the ambient background. And so it's trying to learn which is which according to a probabilistic model of the data. And that involves sort of training and internal representation of the data for many epochs. So the question refers to the fact that during this training procedure, you hope that the loss function that you're using to train this thing sort of decreases smoothly over time and plateaus and converges. And what can happen on some data sets is, instead of being just a smooth function, you can see some spikes in that. And you don't want that to happen. So as a technical detail, what you can try to do is reduce the learning rate in order to prevent those kinds of spikes. The learning rate is sort of optimized for the general data set case and works for, I would say, the vast majority of data sets we've seen so far. But there are occasional data sets where it benefits you to reduce the learning rate to try to eliminate some of those spikes. So it is important to take a look at, there's an output PDF that's generated by the tool. Take a look at that. If there are spikes, try reducing the learning rate. And hopefully by tweaking the parameters a little bit, you can reduce those spikes. Let's see, if it looks like, just as a technical, one final technical detail, if it looks like the loss or the elbow as it's written on that output PDF, if it looks like it converges slowly to a reasonably stable value and it's not spiking down at the end, then it should be okay to use. But I would suggest trying to reduce the learning rate to get rid of those kind of spikes in general. I hope that answers the question.
Thank you, and to the attendee, it sounds to me that it does, but if you have another question or a follow-up, please, of course, post that too. Thanks.
Thank you. Okay, so yeah, happy to take any more questions about that, especially about Cellbender, which I know a little bit more about than most of the other topics here. So
I will then jump in with another one that I think pertains to Cellbender and the count matrix more generally. There was a set of questions last time about how to, and at what stage in the process, to deal with the abundance of mitochondrial reads, and whether that's something that needs to be handled on the the pre-data analysis, the experimental end, or at the filtering step, or some combination thereof.
Yeah, that's a good question. So let's see. The idea of Cellbender remove background is basically that it will remove as much noise as it thinks it can explain based on its internal model of how this data is generated. So by that I mean that the output data that you measure is explicitly a sum of the counts in cells and the ambient background counts. Now, how does the tool know what those ambient background counts are likely to be? That is why the empty droplets need to be included as input to this tool. The tool tries to come up with a self-consistent understanding of what that ambient background profile looks like based on what's in all of those empty droplets in that long plateau of the UMI curve. So I don't have a picture of it up here right now, but last time we took a look at the UMI curve, we could see that there's a plateau for cells where the number of reads per droplet is quite high, and then it drops off, and there's a long tail, a long plateau of empty droplets, which maybe have 100 or so counts per empty droplet. So if those empty droplets contain a ton of mitochondrial reads, for example, then the remove background tool will say, okay, I would be justified in removing a lot of mitochondrial reads from the data set because a lot of mitochondrial reads come up in the empty droplets. However, on the other hand, if that's not consistent, if it's not consistent to say, let's remove a lot of mitochondrial reads because, for example, there are not a lot of mitochondrial reads in the empty droplets, then mitochondrial reads won't be removed, might not be removed at as high of a rate from that particular sample. So what I'm trying to say is it's a very sample specific thing. There are some experimental preps which just seem to produce cells with a lot of mitochondrial reads, whether it's because it's a tissue specific thing like cardiomyocytes, which are very metabolically active, other types of muscle, very metabolically active tissue, lots of mitochondria, lots of mitochondrial reads. So if that's the case, then it's possible that a lot of mitochondrial reads really belong in that data set, and cell-bender remove background will not remove them all, and you probably shouldn't remove them all during the cell QC procedure which follows. But people often do take into account mitochondrial read fraction during this cell QC procedure, which we talked about last time.
Thank you.
Okay, yeah, so that's the review from last time, but happy to talk about any of it further. So let's see, this time I'll talk a little bit more about a few things that we didn't have time to touch on very much. So that is the question of what happens if you have multiple data sets. Specifically, the difficulty here with multiple data sets is what becomes apparent very quickly if you try to just analyze these things by smashing them together without doing anything special, is that you're going to run into batch effects, and it becomes important to understand how to handle those. So here from the Butler et al. Nature Biotech paper about Surat is a figure that shows a little bit about the scenario of several data sets. So for example, you might have two data sets, one in the presence of some drug, one as a control without. You prep your libraries and sequence things separately. For example, you do your data analysis, and what we've talked about is just data analysis for one sample. So that procedure we discussed has to happen for each sample individually, just totally separately to get this count matrix. And then we have several count matrices, and the idea is, what do we do with these several separate matrices? How do we combine this? If we just concatenate the cell dimension together and continue with our map making procedure, we'll end up with something that looks like this, what's called here an unaligned data set. And you can see that there are two colors this red and blue sort of representing the cells that come from these two conditions, treatment and control. And so this is a bit confusing, like without batch effect correction. Is this really what's happening? Are there 5 different cell types? 2 of them are brand new for the drug-treated condition, and 3 of them appear only in the control, and there's no overlap? Well, it seems unlikely. So what's really going on here? And it seems that what's really going on is there are batch effects. There are some technical details which could have a variety of potential causes that preclude an interpretable analysis. So the Surratt version 2 paper suggested a procedure to help with this. And that procedure results in the following sort of a workflow where you have an unaligned data set first, where you've just concatenated together your count matrices from different samples. And then you perform some kind of batch effect correction procedure, which results in an aligned data set that looks something like this. Now, where we're coloring by batch or equivalently by condition, treatment versus control. And we can see that within these blobs, which represent clusters of cell type, we can see that there's a mixture of both conditions, treatment and control. And so now we hope that what will be the case is that when we do this downstream analysis of clustering and visualization, we'll see that we pick out maybe 4 different cell types, and the interpretation now is much more clear. There are 4 different cell types, each of which had a representation from treatment and control groups. And we don't actually believe that this unaligned data set is very informative, where it sort of looked like everything was very different and it was hard to tell what was going on. So the process of batch effect correction really enables us to interpret our data. So how does that little arrow from the last slide, how does that happen? There are sort of 2 schools of thought on how best to do this. As I mentioned last time, the first is to group the cells together, but don't alter the count data. So the procedure just involves modifying that low dimensional representation of the cells, that dimensionality reduced representation, so that the cells will group together in a graph, for example, but not altering the raw count data. And then when we do downstream differential expression testing, we can model a batch variable, for example, during that testing to give us accurate differential expression results. The other school of thought is to use a method which actually manipulates the values of the count matrix itself so that the data sets align. And then downstream, during differential expression testing, you can just use these manipulated values of the count matrix directly, and you don't have to worry about modeling in something for batch later on. However, I personally am a big fan of the first school of thought, where you use a batch effect correction to group the cells together differently. You can modify your low dimensional representation of the cells, but not alter the actual count data itself. And then to later model batch during statistical testing. I sort of think this is a little bit more conservative of an approach, and this is the one I tend to stick to. And lots of tools provide an option to do it this way. So specifically, those software tools, I'll highlight 3 of them here and really focus on 2. One of them that I like a lot is SCVI, stands for Single Cell Variational Inference. The GitHub repository is listed here. This is a tool that sort of similar to the cell bender remove background approach, uses a probabilistic generative model to try to account for the effect of batch on the data set. So it tries to learn a low-dimensional latent representation of gene expression per cell that is independent of batch. So that's scVI. The second tool that I'll mention is called Harmony. There are a couple different examples of implementations of this general idea of harmony. And the one that I like a lot, because I'm more of a Python person than an R person, is Harmony PyTorch, which has been implemented by Bo Lee's group. And the GitHub repository is listed here. So these are both quite useful. I'll talk a little bit more about each of them. Seurat version 3, if you're a fan of tools written in R, it is also quite useful. It has its own method for doing batch effect correction, which does seem to work pretty well also. So that's another option. Personally, I'm a fan of these first two a little bit more. So I'll mention what these two do. This scVI tool, you can start with raw count data, which is one of the things that I like about it. You don't have to do very much upstream processing. Start with raw count data. You also should do highly variable gene selection as well. So as we talked about a little bit last time, selecting highly variable genes is an important part of the whole dimensionality reduction procedure. So into this scVI tool, you can feed in raw count data for the subset of genes that is highly variable. That's a good way to go about it. And after running, this tool will produce for you a set of low-dimensional latent variables. So it'll produce a vector of maybe 20 or 50 elements, depending on which parameters you choose when you run the tool. And you will have a maybe 20 or 50 dimensional vector for each cell. And those 20 or 50 numbers will represent the gene expression within each cell. And they behave sort of like batch effect corrected principal components. So you can use them downstream for running UMAP or doing clustering, for example. And this is a picture that's been taken from the scVI paper that shows sort of the effect that this batch effect correction method has. This first column shows PCA only. This is in effect no batch effect correction. So here in this top left panel, we're coloring the cells by batch, and we can see that the data is separating mainly based on batch, not based on cell type. In fact, if we color by cell type, it's rather confusing to see where the groups of cells are. In contrast, if instead of just doing PCA, which is like no batch effect correction, if we use this scVI tool, we can end up with these sort of batch effect corrected principal components, which we can use to make a map. And that map, if we color it by batch, it looks like this, where these clusters are really well mixed in terms of which batch the cells came from. And that kind of mixing is sort of what we would expect to see if these are experiments that are repeated samples of the same type of tissue or treatment and control, for example, where we expect those things to be pretty similar.
Stephen, there's a question about what to do after you have the clusters. And the question is, once you have the clusters, can you recommend a tool for cell marker identification? There are a number of maximum likelihood approaches, but what if the cell counts are very low, say 200 to 700 cells? The maximum likelihood approaches may not be as useful, and there may also be limitations in terms of sample and tissues being tough to get in abundance.
Yes, that's a great question. And it does turn out that some of the canned methods for picking marker genes, like for example, just going ahead and doing a Wilcoxon test, which is one of the methods that are available from several of these tools like ScanPy and Seurat. Oftentimes that is not the best approach to take, and so I will cover in a little bit of detail my recommended workflow for doing differential expression testing. And so marker gene selection is one particular differential expression test that you can run. So it falls under that heading of differential expression testing. And hopefully I'll cover that in just a couple slides.
Great, thanks. And just so that I don't create confusion, the person who posted the question has clarified that when I said maximum likelihood, I actually should have said machine learning. So just to avoid conflating those two terms.
Oh, okay. Interesting. Yeah. Okay. Yeah. Yeah, so I'll cover that in just a minute. But yeah, so this scBI approach, you can see that now the data is separating by cell type, and that's the intention there. So similarly with this approach called Harmony, an approach that is very different in the technical implementation, but also seems to do a very good job. Again, check out the Harmony PyTorch implementation, especially if you're a Python-oriented programmer. This takes PC principal components that you've calculated and batch effect corrects them to produce batch corrected principal components. So you do need to start with principal components here as opposed to raw counts. But getting these principal components is something that most people are very used to, so it doesn't present an issue. And this is just from the Harmony paper showing before and after. Here they're actually looking at 3 different chemistries of library preparation and showing that they're totally separated without doing this kind of integration or batch effect correction. And after the integration procedure, something like this, where these different chemistries or batches are mixed and we can see the different blobs are different cell types. One thing I'll mention is that this Harmony approach is very fast and scalable. The scBI approach, while I do like it, and it does scale quite a bit, it takes a couple hours to run. And in my experience, if you try to go up beyond like 750,000 cells, somewhere in that area, you can run into a problem. So a lot of times people now are working with these big atlases, integrating a lot of data sets. And so if you want something quicker that can scale to 1 million cells and beyond, although scBI may be able to handle that in the future, it has a hard time right now. So Harmony is a good bet. And it's also very, very, very fast. You can run an integration of maybe 20 data sets and have 80,000 cells. It'll run in maybe a minute. So it's very, very quick. This is far from a solved problem. Of the 3 methods that I've mentioned, 1 is from 2018 and 2 are from 2019. So this is a rapidly evolving field. There's been a lot of progress made recently. It's also kind of a fun computational problem. So a lot of people are working on it. And there are many, many more methods out there, many of which I haven't even tried. So important to keep up with the literature and see what new methods are coming out. And just before I get to differential expression, I'll mention subclustering because it's a task that a lot of people want to do, but it's often kind of ill-defined. So I'll just mention that the task of subclustering depends upon many things, and it's kind of a nebulous task in that the idea of subclustering is to say, For example, let's say I have this result from Harmony. I have my batch effect corrected clusters. Now I want to say which groups of cells belong to the same cluster and which are different clusters. It's almost bordering on a philosophical question here in some regards. This blob that's over by itself, maybe that's a unique cell type that's different from these cells and these cells, and that's fine. That's pretty interpretable. But for example, this cluster here, is it one cell type that has some variation within it, different cell states, for example? Or is it different cell types that just happen to be kind of similar and there's sort of cells in between on a kind of continuum of expression? So the question is, when we do clustering, what resolution do we go to? Do we try to split this up into maybe 1, 2, 3, 4, 5 groups? Or do we leave it all as one big cell type? So as I mentioned, it's almost a philosophical question here about what defines a cell type. Is it a phenotype? Is it a transcriptional state? How variable can that transcriptional state be in order for the cells to be classified as one cell type? There's no great answer to this question, but for example, if your strategy is to leave this whole group as one broad cell type, then one downstream task you might be interested in is, okay, if I cranked up the clustering resolution a little bit so that I got maybe 1, 2, 3 clusters, say, well then what would the difference be between those? And those would be subclusters at that point. So that's the subclustering problem. And so I'll point out a couple of the little pitfalls here that are involved with subclustering. So for example, this is a little bit of analysis that I did as part of the precision cardiology lab, where I won't give a lot of details, but we were looking at fibroblasts, for example. This was all the fibroblasts from one particular set of experiments. So this was one sort of large cluster. And what we could do was we wanted to look at maybe subtypes of fibroblasts, look and see if there were any specialized fibroblasts doing different things. And so if we turn up the clustering resolution, we can divide this one big blob into several smaller groups. But it's often hard to interpret what these groups are. And one thing that's important to keep in mind when you're doing subclustering is to be careful about how you quality control cells, because it can make a huge difference. In this case, when we did strict elimination of doublets using a tool like Scrublet, and using harsher mitochondrial cutoffs to eliminate things that we thought potentially were dying cells or lower quality cells, we ended up with a totally different UMAP representation of the data, which was quite a bit more interpretable, where these clusters now are, these subclusters are very different from what we would have gotten just by naively doing this without carefully QCing the cells further. Okay, but to get to differential expression testing, this is really the most fundamental question that we can ask of the data. And for example, it's part of selecting marker genes, as the previous question mentioned. So once we have these clusters of cells, we want to see what's different between them.
Stephen, just before you start that section, there was one more question about Cellbender. And the question is whether it helps to remove doublets or whether one should use Scrublet to do that afterwards.
I would recommend having Scrublet do that afterwards, yeah. The Cellbender remove background tool should be able to handle the presence of doublets just as if they were their own cell type. And for the purposes of that kind of background removal, it doesn't matter. It doesn't seem to matter too much whether something is a doublet or whether it's just another cell type that looked like a mixture of those two different expression profiles. In practice, it doesn't really help to remove those doublets beforehand. And so it's safe to just feed in the whole raw count matrix without doing that kind of pre-processing, and then try to remove the doublets afterwards.
Great, thank you. And I will also just interject that sadly, we're once again coming close to time. We probably have about 3 minutes left. And thank you so much for your willingness with all of the interruptions. And maybe there are a couple more minutes to continue, just to let you know.
Okay, sounds good. Yeah, so I mentioned this a little bit last time, so it's not completely new, but recipes that I would recommend for differential expression testing include this one here. So if you have a chance, check out this paper. RNA-seq analysis is as easy as 1, 2, 3 with lemma, glemma, and egar. This is a good paper that has really the nuts and bolts details spelled out about how to use these tools, a lemma-based approach to do RNA sequencing differential expression analysis. This paper is really focused on bulk RNA sequencing data. And to move into single cell RNA sequencing data, I'd recommend that you take a look at this paper by Lunn and Marioni, Overcoming Confounding Plate Effects in Differential Expression Analyses of Single Cell RNA-seq Data. The idea here is to take this RNA-seq approach and make it applicable to single cell data by summing your single cell data over groups of cells that are in the same condition. And so that depends exactly what we're talking about. But for example, let's say we're doing a treatment versus control study with a number of different cell types and a number of different individuals. This kind of summation to get pseudo bulk measurements might involve summing over cells per cluster per individual, for example. So dividing the cells up by cluster and by the individual person or subject that they came from, we can create a pseudo bulk vector of expression for all the cells from that individual, from that cell type. And so now we have the single cell information retained because we're not summing across different cell types, but we have a pseudo bulk expression vector which we can use in this kind of lemma and VUM analysis framework. So I really recommend checking out those two papers. Yeah, as I mentioned, this approach involves summing to get pseudo bulk measurements. That's this paper here. Handling count data's heteroscedastic nature using this approach called VUM, which is detailed in this paper, and using a linear model in general to account for fixed effects. Lemma is the tool to do this kind of linear modeling, and this is the associated paper. So accounting for fixed effects, what do I mean? We can write down a linear model here in this R syntax. It might look something like this, where if we're testing for marker genes, we might put together a model that looks something like this, where our output counts that we observe have some fixed effect due to the cell type, the cluster. They have some fixed effect due to batch, and this is the point where we're modeling in batch during our differential expression test, as I mentioned previously. And maybe male or female, so we can contrast one cluster versus others. We can pick out those particular contrasts which correspond to one cluster versus all others, and those can help us find log fold changes and p-values for all these genes which correspond to marker genes for one cluster. But for example, using this linear modeling approach is powerful because you can write down any number of models for the data set and design of your particular experiment. So if you have different tissues, different treatments, for example, different individuals, you can account for all these kinds of things in this linear modeling approach. So it's flexible in that way. Oops. And just to mention, this particular sort of recipe that I'm recommending, Lemma plus Voom, comes out looking pretty good in a recent review comparing all these differential expression methods for single cell RNA sequencing. So I believe that it's a pretty good approach. And with that, let's see. I think I will, let's see. Basically, I'm done here. I just want to mention that when you're doing experimental design, it's important to keep batch effects in mind. Try not to do all the healthy samples in one prep and sequencing and all the disease in another prep and sequencing. Combine things so that you can mitigate the effective batch by comparing within groups.